last 5 or 7 years on translating these really interesting insights we hear in model organisms from network biology into precision therapeutics, specifically for cancer, which is what my lab works on. So the title of the talk is about tyrosine kinases and using the interaction map that we defined to identify a new network state that we think exists in lung cancer that is targetable with small molecule inhibitors of this kinase EGFR, which I'll tell you about. So my lab, we're very interested in signaling and systems aspects of signaling. In this study, we're very interested in receptor tyrosine kinases. And this stems from our understanding about receptor tyrosine kinases as being these principal sensors for outside signals in the cell and then transmitting these into the inside of the cell into activating different signaling pathways. This is a very standard picture you'll see in molecular biology of the cell or any textbook. But as you can expect with anything in biology, we feel that these pictures are much too simplified and they don't really reflect the complex reality of what we see in any cell in the body. And the way that this protein or this class of kinases is typically diagrammed is through this very simple diagram here where you have ligand binding on the outside of the cell, which then results in dimerization of 2 receptor tyrosine kinases. This results in transphosphorylation of the C-terminal tails of these proteins on the inside of the cell. This then serves to recruit different adapters, which then activate various signaling pathways. This is really the dogma in the field, and this is kind of the way that everyone thinks about the simplified diagram. But really, this belies this largely complex and newly discovered, in many cases, biology complexity of the signaling. These particular proteins are under constant control. They're constantly being recycled. They're constantly being shuttled to the cell surface. They're constantly being degraded, and they're also constantly being turned on and off by different phosphatase activation. And then other kinases can actually come in and activate these too. So these proteins, while we thought they were quite simple, actually exist in this very complex ecosystem. Now, likely because of this very stereotyped understanding of receptor tyrosine kinases and the way that they signal, we know very little actually about the sort of interaction landscape of this family of proteins. So there are 90 human tyrosine kinases, 58 of which are receptor tyrosine kinases. And what I've just underlined here in these sort of big dots here is the amount of network knowledge that we have in public databases around the physical associations with these proteins. And you can see it's really centered around just a handful of these proteins. So why are we interested in this family? Well, as you may or may not know, this is a central family of proteins involved in kinases, involved in cancer. And really, these are thought to, a number of these proteins are thought to be drivers of different types of cancers. And I've just listed here the ways in which they're activated. And really what the field is fixated on is on the activation of these kinases through genetic changes in cancer cells. For example, you can have amplifications in the kinase HER2. And this is then FDA, a breast cancer which has the presence of HER2 amplification is then eligible for treatment with HER2-directed antibodies. Mutations in a number of these kinases is then paired with different small molecule inhibitors, and then also fusions and rearrangements of these kinases also indicated for treatment with different types of small molecule inhibitors as well. And so this is really the dominant form or dominant mechanism of oncogene activation in cancers, and most cancers, I would say, have some sort of mutation in this pathway. Now, in my lab, we've been interested in other ways that these kinases can become active. And beyond just thinking about genetic mutations or genetic changes, we started to also think about how they could be hyperactivated in their wild type forms. So this is not sort of point mutations, but actually just elevated levels of these proteins. And so if you look through, this is from the TCGA studies of 10,000 or so tumor samples, you can find that across this family of protein kinases, many of them are, one, amplified in many different disease types, but there are also many of them are overexpressed in different disease types. And so this kind of piqued our interest in this family because this suggests that there's hyperactivation of the wild type forms of many of these kinases, not just the small number that we've all studied. So we set about a number of years ago in collaboration with Nevin Krogan at UCSF to clone and tag and systematically affinity purify all 90 human tyrosine kinases. And this was done in HEC293 cells with a sort of a large number of replicates consisting of over 400 APMS experiments. And so putting these data together, There we go. We obtained mostly uniform and sort of unbiased survey of the physical interactions of all kinases in this family. So what do we use this for? So first we took the data, and as is the standard in the field, we compared it with other data sets. So for example, here, this is looking at our APMS data scored with what we found is the most robust scoring system, CompassZ. And what we looked at is at different thresholds of our network, what is the overlap with protein-protein interactions deposited in IRIF-Web. That led us to think that this sort of region, this cutoff of about 2.5 was a reasonable cutoff. And then similarly, if we look at overlap of our network with known phosphorylation kinase and substrate pairs, we found a similar peak around 2.5. And so we thought that 2.5 was a reasonable cutoff here to take. Principally at this cutoff, then we were able to identify interactions for all 90 human tyrosine kinases, which we think is something that hasn't really been explored much in the public databases out there. There's only been sort of partial analysis of this family. So this is the sort of unbiased map that we generated. So it's obviously quite a bit of data, and so we spent some time trying to figure out what the best way to look at this data is. And what we reasoned was that if we could identify interactions that cluster, that the interactors themselves are known to interact often in known protein complexes, then we can identify sort of key salient features of biology that were being perturbed by this family of proteins. So here in these colored squares are the tyrosine kinase, and then here using sort of a very simple algorithm looking for triangles and combining all these triangles where we have interactions with the APMS data in red, and then blue are literature-based interactions from quorum or other curations of different interactions in the literature. Then we look for these triangles, and we merge all these triangles together to identify this map. It's somewhat curated, but also somewhat systematically prepared as well. And what we identified here is that a number of these kinases then interact with known protein complexes, which was really surprising to us, specifically because most of these connections hadn't been described. For example, here, I'll tell you this about a little bit later. Here's two receptor tyrosine kinases that interact with this transcriptional elongation complex. For example, here we found that SARC, which is one of the earliest oncogenes identified in cancer, interacts with this lysosomal ATPase and the regulator complex, which contains mTOR components as well. So these are sort of new connections that piqued our interest, not only because they came up sort of in this unbiased survey, but they're also corresponding to interactions that span multiple components of the same protein complex, right? So we don't think these are sort of false positives because we can find multiple members of the protein complex coming down with any single tyrosine kinase. So this is sort of a survey of these complexes. So these are quorum complexes that we pulled from the literature, and you can see that many of these tyrosine kinases that interact with these complexes. Here's some of the more close-up pictures, and I'll tell you a little bit about some more of these in a minute. So when we look at these complexes, though, one thing that really stood out to us was the fact that many of these complexes aren't localized, for example, at the plasma membrane. Many of them are actually nuclear. So for example, PTEF-B is a transcriptional elongation complex, which is a decidedly nuclear complex. And many of them have other localizations too, for example, on the eichmann cytoskeleton, or at lysosomes, for example. And so that got us thinking that in the pathogenic case where these receptor tyrosine kinases, which is what really drove our interest in this sort of phenomena, in this case where we have overt overexpression of transmembrane receptor tyrosine kinases, how can it be that they actually interact with nuclear proteins, right? This is seemingly a mislocalization of a very strongly plasma membrane bound protein. So to try to understand if this was actually a relevant phenomena and something that's worth studying further, we started to take a look at this human protein atlas, which uses antibodies directed against these particular proteins and then identifying their localization within different types of cancer cells. And so using a very simple guilt by association analysis of the interactors of each of these kinases, we then predicted their cellular localization based on the known localization of their interacting partners. And we found that many of them, for example, the ones shown down here, interacted with many nuclear proteins. And so they're, for example, enriched for interactions with nuclear proteins. And then we asked how many of these are actually consistent with what we find in the human protein atlas. And we can find that many cases actually they were consistent, which was surprising to us because these are transmembrane proteins. And so, for example, here's one, DDR1, which is a receptor tyrosine kinase. We found it interacting with DNA topoisomerases and the TF2H complex and RNA splicing machinery. These are all sort of nuclear or nuclear proximal protein complexes. If you take an antibody, a primary antibody against DDR1, and you stain MCF7 breast cancer cells, you can find that this protein actually indeed does have two lives. It has a life right at the plasma membrane. You can see it sort of surrounding these cells. And you can also see that it has localization, nuclear localization. Sorry, it's a little bit kind of hard to see here, but this is, you can see this sort of dual localization. Similarly, with EPHB6, Ephrin B6, you can find this interacting with chromatin remodeling proteins. And then you can see that and with a primary antibody against this in a cancer cell, you can find that it's localized in the nucleus as well. And similarly with DDR2, it binds with a number of components of the actin cytoskeleton. And in BJ fibroblast cells, it's localized to the actin filaments as well. So this says to us that it's really not that far off, and that we should potentially consider non-canonical localizations of these proteins, particularly in cancer where they're highly overexpressed. And this is sort of our efforts to try to understand more about the biology, the particular complexes and their perturbation states. I won't delve into this for the sake of time, but what I will say is that we found this interaction between FLT3 and this transcriptional elongation complex PTFB, and we were able to validate that this interaction does exist within cancer cells. It's also dependent on the binding of PTFB to chromatin. So if we knock PTFB off of chromatin, with BRD4 inhibitor, we can actually disrupt this interaction. So normally this interaction exists, but then if we disrupt this binding to chromatin, we can knock it off. And it's also functional in terms of activating the CDK9 component of this complex as well. So then armed with this sort of information about the sort of basic biology of these complexes, then we started thinking about how can we use this understanding of the network of tyrosine kinases to enhance sort of treatment of different cancers. And really, we focused our efforts on lung cancer for a variety of reasons. One of which is that tyrosine kinases and their mutation this is really the poster child for precision medicine, and one where tyrosine kinases are the dominant drivers, or this tyrosine kinase pathway, which is signaling through tyrosine kinases, then through KRAS and BRAF, and ultimately the MAP kinase pathway. This is a sort of central axis of mutation in these lung cancers. Now, critically, about 25% of lung cancers have no known mutation in any oncogenes, right? So this is a case where we've done really well. We have a number of therapeutics, and we have a number of sort of diagnostic tools to try to diagnose these tumors. For example, if a tumor presents with an EGFR mutation, and it's treated with EGFR inhibitors, we see often very significant responses, initial responses to these EGFR inhibitors in lung cancer, about a 60% response rate. But however, these oncogene negative tumors, or tumors that don't have mutations that are currently druggable, they don't benefit from such precision medicine approaches. And because of this selection for this pathway, we thought this would be a case where the networks might be able to tell us a little bit about what's going on, and with our particular interest in these oncogene negative tumors. So there's, like I mentioned, because of the preponderance of mutations of this pathway, we know that there's this tremendous selection for activation. So you can have mutations in receptor tyrosine kinases, you can have mutations in KRAS, you can have mutations in the negative regulator of this pathway, NF1, activation in BRAF, and they all sort of stem in this sort of linear pathway, right? So this is really strong selection. And not only is there selection for sort of generic mutations in this pathway, the different alleles are actually selected for as well. For example, with EGFR, the predominant mutations are in exon 19 and exon 21 mutations. In KRAS, there's a number of different alleles, the most common being G12C, but there's a number of different alleles even at the G12 locus and the G13 locus. And similarly with BRAF, there's also selection for different types of mutations in BRAF. So the most common mutation in BRAF that you'll hear about is these class 1 mutations, this V600E mutation, which is commonly found in melanomas. And that signals it's a strong kinase activating mutation, signals as a monomer. But these other mutations, for example, in lung cancer, the majority or almost half of BRAF mutations are actually class 3 mutations. These are weak activation of the BRAF kinase, and they actually are not sufficient to signal without dimerization. So there's this different sort of allele-specific types of mutations we see in lung cancer. So there's this sort of harmony that has to happen in a lung cancer selection of different mutations. So we hypothesized then that this sort of harmony, or this sort of balance of all these mutations, And activation of this pathway potentially in oncogene negative cancers could happen, not through activation of this particular kinase by mutation or by amplification, but rather by changes in the interacting partners of these kinases. So different levels of these particular interacting partners could change the balance of how much protein is, for example, displayed on the cell surface, or how much of this protein is signaling or competent to signal in the cell. So just the initial test of this hypothesis was first to look for chemical genetic interactions of these interactors. So for example, for EGFR, we took an EGFR mutant lung cancer cell line and took the interactors and knocked them out systematically one by one, and then asked how they alter treatment with an EGFR inhibitor in lung cancer. And what we found is when we did this with EGFR interactors, ALK interactors, MET interactors, or HER2 interactors, the majority of interactors that we found were able to actually alter the sensitivity to small molecule inhibitors against those kinases, suggesting that there's sort of a functional interaction there between the interactions we found and the presence of mutations and activations of these oncogenes. So then we started to think about how this network could be used sort of en masse to help diagnose cancers. And so this is, I think, the sort of the overview of the approach here. So here what we did is we first took all of our network data and also the network data that's been published in the literature around these kinases. And then we combined it with tumor gene expression profiling data, and it found the interactors that were differentially expressed when we knew that the particular kinase, in this case EGFR, was activated by mutation. So this has ended up with a subset of our network. And what we found is this network, I believe this is about 27 interactors. We found that the expression of some of these interactors was positively linked, means their expression in tumors at baseline before treatment was positively linked with the presence of a known canonical activation of EGFR. And expression about half of these was negatively linked, meaning they were lower in tumors with EGFR activating mutations. And so when you look across around 400 different lung cancers, this is the expression of these network components. And then this is the presence of the mutation in these lung cancers. And then this is our classification of what we're calling the EGFR network state. And so this network score is really a quantitative score, but we can cut it off based on different confidence intervals to say that this is a particular network positive, EGFR network positive tumors. And one thing you'll notice is that most of these EGFR network positive tumors are EGFR mutant, which is by design. You can find that some of them are KRAS mutant, which is interesting because KRAS mutant lung cancer is actually contraindicated for receiving EGFR inhibitor therapeutics right now. And then many of these tumors you can see are gray in both, meaning that they have no known activating mutation in KRAS or EGFR, right? So there's a sort of diversity here of different types of tumors, but you can see regardless of the mutation status, they all have upregulation of these positive network components, and they all have downregulation of these negative network components. So together their network activity is high. So overall, 95% of EGFR canonical mutants have this EGFR are network positive. This is sort of by design in the algorithm. But when we have a sort of a variant of unknown significance in EGFR, about 50% of those cases are EGFR network positive. So this helps us potentially understand what the function of these variants of unknown significance are. And then for EGFR wild type lung cancers, we found about 20% of those are network positive. So the question then becomes, what is going on here? Are these network positive tumors, are they actually signaling through EGFR or not? And so this is what the networks look like in our case. So for example, this is one particular patient tumor. This is the EGFR network in an EGFR mutant lung cancer. And you can see a bunch of these positive components are upregulated. For example, ALB and these other ones are upregulated. EGFR itself is not upregulated, and these these negative regulators are downregulated. In contrast, an EGFR wild type tumor with a very low network activity score has the flip of this network state. Now, down here are the more interesting cases. These are EGFR wild type, but they have very high network activity. For example, you can see this upregulation of these components. You can see high expression of EGF, which is the ligand for EGFR, and you can see high expression of EGFR itself, and then sort of no expression in the negative components. And then similarly here, this is a case where you don't have any changes in EGF or EGFR, but other components of the network are activated. Right. So although these are wild type tumors, their gene expression or the network state seems to indicate that the EGFR pathway is active. So what evidence do we have that this is actually functional? Well, I think here is one of our first indications. When we look at the phosphorylation of EGFR, so this is the activation of EGFR commonly through dimerization. What we find is that these tumors with higher network activity have often higher phosphorylation of EGFR. And this happens also in the cases where EGFR is wild type, right? These gray cases as well. So that suggested to us that this is indicating tumors where EGFR is activated by phosphorylation, independent of mutation. So to put this sort of model to the test, we wanted to understand if EGFR was activated in these wild-type tumors, are these then tumors susceptible to EGFR small molecule inhibitors? So to first study this in a sort of a retrospective way, we assessed a panel of 25 lung patient-derived xenograft models, these mouse models of lung cancer developed by Novartis. And each of these dots indicates an independent tumor from a different patient implanted into a mouse, then treated with an EGFR inhibitor, in this case, erlotinib. And if the tumor grew after treatment, you would have, that's these cases here, where the tumor doubled or doubled, tripled, or quadrupled after treatment. So these are progressive tumors. And the tumors in green then are the ones that responded or shrank after EGFR treatment. Only one of these tumors had an EGFR mutation. They had a non-canonical mutation in EGFR, which is a variant of unknown significance. And a number of these are also KRAS mutant tumors. And these are tumors where that normally their patient counterparts would be contraindicated from receiving EGFR inhibitors. And so you can see this correlation here where a number of tumors did actually respond to EGFR inhibitors. Those tend to have higher network activity. As a control, this EGFR variant of unknown significance had the highest network activity and had the most regression after treatment. And you can see these mice that had these tumors that had a higher network activity scores before treatment, they tended to live longer because their tumors didn't grow as much. And this sort of phenomena is very similar to what we see in cell lines as well. So for example, here, this is a panel of KRAS mutant lung cancer cell lines. So these would never, again, not get treatment with EGFR inhibitors, but you can see that their sort of IC50 or their sensitivity to the EGFR inhibitor erlotinib is predicted by EGFR network activity as well. And we've seen this with erlotinib, and we've seen this with another cohort with dacomitinib, another EGFR inhibitor produced by Pfizer. And some of this work was done in collaboration with Pfizer as well. So since I'm running out of time, I just want to sort of get a couple of points across here. So one is that we were trying to understand how useful is this network for this actual exercise? Like if you just had a gene expression signature, could you find the same thing? And so what we did is we tried a number of different supervised machine learning approaches. We used gene expression signatures and then a bunch of regression-based approaches. And we assessed the same performance on the same sort of benchmark validation exercises that we did. So the sensitivity to erlotinib or lapatinib, the sensitivity of these patient-derived xenografts to erlotinib. And what we found is actually the network approach is actually the only one that was consistent across all of them and giving roughly the same or better performance. And so it seemed to be a more robust measure of network activation or EGFR pathway activation, and one that could replicate across 3 different platforms. And just thinking about then how these mutations generated? Like, why does this actually happen in these lung cancers? Because you would assume that the easiest way to activate this pathway would be through mutation. So why would a tumor then go through the trouble of activating this network if you accept the argument? Well, what we think is actually happening here is that the majority of these tumors are actually oncogene negative. So they don't have EGFR, they don't have any mutation in an oncogene that we know of. So that's going to our goal of trying to decipher what the biology of oncogene negative tumors are. But many of these tumors actually have a mutation in NF1, which is a negative regulator of the MAP kinase pathway. And to our surprise, many of them also had KRAS mutation. NF1 was the most enriched in this cohort. And so NF1 is a sort of a negative regulator, but it's also a weak activator of the MAP kinase pathway. Similarly, the BRAF mutations we see in these tumors are enriched for class 3 mutations. And the KRAS mutations we see in these network positive tumors seem to be specifically G12A mutations, which is reported to be one of the weaker alleles of KRAS to reduce RAF binding. So we reason then, based on these data, is that this is actually a dual driver situation. So in most cancers, you have one strong blazing driver of tumor growth. And that's the one that we all know about, and that's what everyone's looking for. But in these particular cases, what appears to be happening is that you have one weak driver of tumorigenesis, for example, NF1 mutation or a class 3 BRAF mutation, and you have a second event, which is this activation of the EGFR network. And those two together are sufficient to drive tumorigenesis. And so for the purposes of time, what I'll show you is our efforts to try to validate this model. And so we were thinking about how we might be able to use this approach as a way to treat these oncogene, mostly oncogene negative, but EGFR network high tumors. And we did 2 prospective studies, animal studies. So one is we collected a bunch of cell lines that have these non-canonical, they're wild type in EGFR, but have these mutations in NF1 or G12C or G12A mutations or BRAF class 3 mutations. We did confirm that treatment with erlotinib was sufficient to reduce the flux through the BAP kinase pathway, which means that these tumors, even though they have these mutations in these oncogenes, they still respond to EGFR inhibition at a molecular level. And then ultimately, when you treat some of these tumors, in this case, a G12C mutant tumor with EGFR inhibitor, you also see a response in the tumor growth. So the last piece of validation I'll tell you about is this collection of PDX models. So we work with Champions Oncology to get a list of lung cancer PDX models that were all EGFR, well, almost all EGFR wild type except for one. So this is all the lung cancer PDX models that they have that have gene expression data that we could assess our network score. The top one, as we'd expect, is the EGFR canonical mutation. That one has the highest network activity, just as we predict. But the second highest one here has this BRAF class 3 mutation, just exactly how our model would predict as well. So we took this particular tumor model and then grew it in a mouse too, and we could see there that erlotinib had an effect on tumor growth in this setting as well. So we think that this is a potential way to find, identify these tumors that have hyperactivation in a particular tyrosine kinase that is targetable, but is not really amenable through standard next-gen sequencing approaches. So clinically, is this actually a feasible concept, right? So, I mean, are we just sort of grasping at straws here? Well, if we look historically at EGFR inhibitors and how they're used in lung cancer, in EGFR wild type and also mutant patients, the response rate in EGFR wild type, mutation wild type patients was around 8%. This is obviously not enough to advance the development of these drugs in this indication. So for example, the response rate in EGFR mutant patients is about 60%. But if you think about what our studies indicates is that a subset of this EGFR wild type patients actually have what we think network activation, that lines up pretty well with this 8% response rate. We estimate it's about 19%. So just to kind of conclude here, the model here is that in most lung cancers, you have a single strong driver of tumor genesis. So EGFR mutation drives this flux through the pathway. This makes up about 12% of lung cancers. But it seems that when you have weak oncogene activation, these particular alleles in KRAS, NF1, or BRAF, that's coincident with activation of the EGFR network. Those two together combine to drive tumor genesis in about 16% of lung cancers. So, so just to conclude here, what we think is going on is there's this tremendous selection for activation of this pathway. Tumors will think about any way that they can do it. And one may be, the most obvious way is through mutation, but the other obvious way could be through changes and alterations of interacting partners of key oncogenes. And this leads to what we think is reuse of EGFR in about up to 16% of lung cancers. And this may be a targeting opportunity that we've missed in the past. So with that, I'll take any questions. Thank you.
You're using this without mutation, or is it without mutation in EGFR?
It's without mutation in EGFR. I kind of went through this for the sake of time, but what we identified was actually there was, I skipped this, and I'm sorry for that. We found that some particular EGFR network positive tumors are enriched for certain types of mutations. So for example, some have RBM10 mutation or RB1 mutation as well. So it's not a mutation in EGFR, but it's a mutation in something else.
And one more technical question. So you have the figure with the xenografts that you treated with erlotinib. So this percent scale axis, what does it mean? Because it's different thresholds.
Oh, I see.
Is it compared to some kind of control vehicle?
So yeah, the way that these studies are set up, it's really like a mouse clinical trial. So for example, with a patient, you never have like a vehicle control. So the way that they set this up is all these tumors are growing at different rates, just like in the human situation. And they're treated with an EGFR inhibitor. And they're just looking for progressive disease, meaning the tumors keep growing, stable disease where the tumors remain flat, or regressive disease where the tumors shrink from baseline. So there's no rolled into this is not an indication of how fast the tumor is growing. But I think because there are PDXs that have been propagated, it's implicit that they do grow. I just don't know the rate at which they
Application. So it's not that group. They were kind of condition dependent. So to what extent the patient state of kinds of comes to play a role in this interaction networks? Are they everything dependent on the say phosphorylation state or whatever?
Yeah.
Or it's just, you know.
Yeah, so that's something we're working on in the lab. So what I can tell you is what we're very interested in is understanding the exact mechanisms by which these network components alter EGFR signaling or potentially EGFR trafficking. Right. Or EGFR turnover. So there's multiple different aspects which these components all different, all change. So there's no sort of one answer. For some of them, we know that they're involved in internalization. Some of them we know are ligands, extracellular secreted ligands that sort of activate EGFR. There's really not one specific mechanism here. We think it's actually many mechanisms. We actually, by doing sort of non-negative matrix factorization on this network state, we actually identified 6 sort of substates. And we think that what we're looking at now is these 6 different substates. And we think that they have different biology. Each one of them has different biology. And that's the slide I missed, and it has come up twice. But for example, there's 6 substates that we identified. One of them is actually seems to be ligand dependent, so overexpression of EGF. But another one seems to be expression of another downstream regulated PIK3R3, which is a component downstream of EGFR. So there seems to be at least 6 different mechanisms that we can pick up.
I think there's been established that 30% of all tumors MYC levels are increased, either through amplification or other reasons. Did you check MYC or MYC-N, MYC-C levels in your system and see whether that affects or contributes to the phenotype you see?
Yeah, MYC, MYC, right? Yeah, we did look. So we looked at MYC amplifications. We actually looked at every, all these tumors have whole exome sequencing. So we looked at all possible mutations that have been reported, and these are the ones that were showing that were actually significantly enriched in any particular type. Thank you.
Meet again at 2:00.